Hi, so this video is going to be about meiosis. In the previous video, we went over a different type of cell division, which was mitosis. One of the goals of this video to show the main differences of meiosis compared to mitosis. So by definition, meiosis is a specialized type of cell division that occurs in organisms that reproduce sexually. This means that this kind of cell division produces reproductive cells. Examples include plant and fungal spores. They are the direct result of meiosis. In animals such as humans, we call those cells either sperm cells, which is produced by the male, or egg cells that are produced by the female. The overall idea is that we're going to start with a diploid cell, and at the end of the cell division, we should get cells that are haploid, meaning they contain half of all the genetic materials. Going into the actual process, meiosis is divided into two sections in which the process occurs over the course of 2 rounds of nuclear divisions. They are meiosis I and meiosis II. The reason for the distinction is because these 2 rounds have their own unique significance in terms of what they create. And we will touch on what those significances are as we go into them. In meiosis I, there are 4 stages. We have prophase I, metaphase I, anaphase I, and telophase I / cytokinesis. So first, let's bring in prophase I. In prophase I, we see this homologous chromosome come in pairs, and if you look at them closely, you can see that they are touching one another. The places where they touch each other, one here, and another one here. There's a purpose for that, and that purpose is for the crossing over. Crossing over in genetics simply means the exchange of genetic materials between the non-sister chromatids of the homologous chromosomes. This exchange process can also be called chromosomal crossover. And this phase is particularly important because the exchange of genetic materials is taking place, and I think it's the most important process to understand. So I'm going to spend a little bit of time pointing out what's really happening during this phase. So here we have the meiotic prophase I. At the beginning of the meiotic prophase I, the chromosomes can be observed as long single threads unassociated with each other. This part of prophase I is known as leptonemma. What happens next is the formation of synaptonemal complex. This protein complex is responsible for the synapses, and its job is to basically unite the homologous chromosomes together. And each pair of the homologous chromosomes is known as bivalent. So we have one bivalent here and another one here. This is zygonemma. After the homologous chromosomes come together, each bivalent, that is the homologous chromosomes, they become shorter, thicker, and the two sister chromatids split and they become visible. So we see these four-membered structure, one here and another one here. We have two homologs, which are these two, one there and one here, each containing two sister chromatids. We call this a tetra, so tetra. And one homolog here, and the other homolog here. This is pachynemma. The next one is diplonemma. This is a significant stage because a crucial event is happening, the crossing over. Again, crossing over is the exchange of genetic materials between the non-sister chromatids. So we see this within the tetrads. The sister chromatids separate what we call the chiasma. So the chiasma is here. The place where the non-sister chromatids remain intertwined. The last part of prophase 1 is diakinesis. The nucleus and the nucleus envelope break down. The centromere are attached to the spindle fibers. The important thing to recognize here is that both sister chromatids of a homologous pair is pointing to the same pole, and that the two homologs are pointing to the opposite pole. So we have one homolog here going this way, the other homolog here going this way. And same thing goes to this pair. One homolog is there, is going that way, and the other homolog is going to go this way. This is, again, diakinesis. Now that we have some basic understanding of what's going on during prophase 1, we move on to metaphase 1. Each homologous chromosome will line up one after the other like this along the middle of the cell. During this phase, notice the chiasma is still intact between the homologous pair. In anaphase 1, the spindle fibers pulls the homologous pairs apart to the opposite end of the cell. And you can clearly see the exchange of genetic materials between the two homologous chromosomes. A section of this chromosome here is pink, and the other chromosome has a little bit of yellow here. Telophase 1. The chromosomes are going to gather at the poles of the cells, whilst the cell cytoplasm divides. Then we get a complete division that gives us these two cells in cytokinesis. Up to this point of the cell division is known as meiosis I. It's also known as the reduction division. In meiosis II, there are also 4 stages, which are prophase II, metaphase II, anaphase II, and telophase II. As we go along the stages of meiosis II, you start to notice that the process is very similar to mitosis. And you'll be correct. And in fact, the processes and the phases are exactly the same as mitosis in terms of how the chromosomes are segregated. So, prophase 2, new spindle fibers form around the chromosomes. In metaphase 2, again, the chromosomes line up along the middle of the cell. The spindle fibers are attached to the protein kinetochore near the centromeres of the chromosomes. And in anaphase 2, the centromeres divide and the chromatids move to the opposite poles of the cells. And finally, in telophase 2, a nuclear envelope is going to be formed around this set of chromosomes and also this set of chromosomes. Then the cell cytoplasm is going to divide this way and this way. And then we finally enter cytokinesis, where the cell is completely divided. So the two cells are going to look like this. Something like this. We also get the same process for the other cell at the bottom here, and it's going to look something like this. And we get a total of 4 cells here that are haploid. And we could call this 4 haploid cells gametes.